filtrate. Once the filtrate enters the nephron, you have both reabsorption, and that's going from the tubes, coming from the tubes, going back to the capillaries, back into the bloodstream. And then we have secretion, and that's from the tubules going into the tubules, or into the nephron itself. And like I showed you before with this slide here, if we take that whole nephron and stretch it, which is the yellow up here, we have Bowman's capsule, which is going to have filtration. Then you got the tube, whether proximal convoluted tubule, loop of Henle, or distal convoluted tubule. If the products are going to go from the tube into capillary, that's reabsorption. If it comes from the capillary and go into the tubules, then we have secretion, and anything that comes out is going to be excretion. Keep those words in mind. You can almost see the types of questions I might say. Making fluid go from the capillary to the tubule, is that filtration? Is that reabsorption? Is that secretion? Is that excretion? That kind of stuff. You should know the definitions. Okay, now going back way early in A&P1, or even beyond before that. Diffusion is going to go from a high concentration to a low. Pretty self-evident about that. Easy for that, right? You have a high concentration here, it just naturally will go to a low concentration. Just will do that, all right? You got 50 students in this room, and you got 10 students out in the hallway. The door is open, which way are the students going to go? 50 students inside, 10 students outside. Which way are the students going to go? Go out, right. Until it becomes 50-50, right? Until it becomes, whatever, 25-25, or 30-30, whatever it is, okay? That's natural. What's not natural is active transport. You're going to go from a low concentration to a high concentration, okay? This is not normal. We're going to have to use some kind of energy to do this, okay? So, using that same scenario, we have 10 students out there, we got 50 students in this room. You open the doors, those students out there are going to have a hard time getting in here. It's not natural. Unless there's somebody who's going to push them in there. We have to use some energy to push them in there, okay? So that's what's going to happen. ATP, which is energy, is going to be required for active transport, okay? Now, substances that actively get transported are these. Reabsorption, sodium, potassium, chloride, calcium, glucose, amino acids. All of these will be reabsorbed by way of active transport. They need energy to do this. We also have things that are going to be secreted through active transport, and we also have hydrogen, potassium. These over here are by way of secretion, but not active. It's going to be ammonia and penicillin, right? Just some things in there, okay? Now, sodium reabsorption. Sodium reabsorption, almost always active transport. Okay? Sodium enters the tubule from the membrane itself, and it gets actively transported out of the tubules by the sodium-potassium pump, also referred to as the ATPase pump. Okay? You guys remember how many sodium go out and how many potassium go in and vice versa? Remember those things? 3 sodium out and 2 potassium in. Okay? My little mnemonic for this, I think I told you this. 3 potassium out and 2. You ever hear that song, na na na? Hey, hey. You heard that, right? Yeah, you see it. Well, think about it. I was in medical school and I was coming up with this. I just heard this song and I was studying action potential at the time. And they were saying it. Na na na. Now, how do you spell NAD? And that was perfect. That's what hit me. I was like, nah, here it is. But listen to it. Na na na. How many times did I say that? 3 times. That's even more perfect. So then I put the whole thing together. Na 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 out. Na 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 out. KK goes inside. And then I just memorized the whole thing. You see? It was much easier now. So I did that for your entertainment pleasure. Don't hear me sneeze. Yeah, that's what I'm saying. Things just pop into me. Yeah. So anyway, that could have helped you in A&P1. I do that with the whole scenario with A&P1 because they do need to know that when you deal with the action potential. But anyway, if it helps you later on in life, super. Okay? Anyway, from there it moves to the peritubular capillaries due to low hydrostatic pressure and high osmotic pressure. Not going to go into that. We kind of did that already with you. We dealt with the glomerulus and the Bowman's capsule. We talked about that. So sodium reabsorption provides the energy and means for reabsorbing most other solutes. Meaning, as sodium comes in, it's using the power of sodium to allow other things to happen. In other words, God bless you, if I'm sodium and your hands are full, your hands are full with books, you're going to use my power to open up the door. See, I'm want to go out, so I'm going to open up the door, and you're going to be able to come in. I'm sodium, and you could be, let's say, I don't know, you could be penicillin or something, and you could come in here, not penicillin, but let's say calcium, whatever. You're using my power to go out that way. Vice versa, there's people out there, they can't come in because they have books in their hands. So again, I'm sodium, I'm going to open up the door. Even though I'm coming out this way, they're coming in this way. You've seen this, right? Antiport, symport, right? That's where you're using some other power to allow you to open up the doors to come in, or in other words, you could be going going out the same direction with sodium. That's symport, or you could be sodium's going out and another one's coming in. That's antiport, right? But it's still a form of like active transport. Okay? Questions on that? All right. Sodium is probably the most common one that provides the energy for other things to come in and out of cells. Okay? Now, let's talk about renal threshold. Renal threshold. Substances, if they're using carrier transports, they can only go in a certain amount. In other words, if this is a proximal convoluted tubule, and I'm just giving you an example. Let's say we have a carrier molecule here, a carrier molecule here, and a carrier molecule here. And here's where your peritubular capillary is. So this is your PCT, and this is your capillary. Now, glucose is here. And if this is a glucose carrier, in other words, glucose could get back in here, but it has to go through a carrier. If we have 3 glucoses here, can all of those go back inside the capillary? 1 for 1. Does it make sense? It can. This one can go right through there. This one can go right through there. So at the PCT, 100% of your glucose will go back in there, because we have an obnoxious amount of carriers that can carry the glucose back in there. Does that make sense? Yes? No? You're not nodding. I'm assuming you know it then, right? Now, here's where the problem is. We have more glucose going through here, let's say for a diabetic. It reaches a maximum. I'm just giving you an example of 3 carriers. There's thousands of carriers, but for my picture. So if I have 3 glucoses, that's enough that these can take these on. But what happens when we have 4 glucoses? Are they going to all be able to get back into the bloodstream? This one can, but this one can't. So what happens to this extra glucose? Goes into the urine, falls right into the urine. This is why people who are diabetic, who have diabetes mellitus, they have a lot of glucose in their bloodstream. They're going to have glucose in their urine. If in a healthy person, we can take on all the glucose in a healthy person because your sugars are not that high, because we have enough carriers to normally take on that. Does that make sense? This is why diabetes. Now, for every molecule of glucose that goes out in your urine, there's 1 molecule of water that also goes out there too, to make sure that the concentration is equal. So diabetics tend to pee a lot because there's a lot of water that's coming out also. Does that make sense? Can you visualize that? All right? So that's what the renal threshold is. It reaches what we call a transport maximum. The amount of carriers here is going to limit what can get into the bloodstream. All right? So it reflects the number of carriers in the renal tubules available and exists for nearly every substance. I just gave you an example of glucose because I think you could visualize that. Okay? And it's there for every substance that is going to be active transport, actively reabsorbed. If the substance concentration exceeds the carriers, then it's going to spill out in the urine. Does that make sense? All right? So like I said, in diabetes mellitus, there's more glucose than what we have as carriers, so they have glucose in the bloodstream. For every molecule of glucose that enters, not the bloodstream, urine. For every molecule of glucose that's in the urine, we're going to have one molecule of water, and therefore their volume of urine is going to increase. All right? Hint, hint, make sure you understand this whole concept of renal threshold and the transport mechanism, or maximum. Is that clear? Okay? So substances are not reabsorbed if they lack carriers. If for some reason we have glucose entering here and there's no glucose carriers, they're not going to get reabsorbed. Okay? If they're not lipid soluble, they won't get reabsorbed, or they're just too large to pass through the pores. Right? We talked all that before when we dealt with endocrinology. The most important non-reabsorbed substances that you should know is urea, uric acid, and creatinine. Okay? And we'll get back to what those are and why they're important. Osmosis, the way water gets absorbed. We dealt with osmosis before. It's just diffusion, going from a high concentration to a low. But what osmosis is diffusion specifically of water molecules. So if there's a high concentration of water molecules to a low, then that's going to be diffusion of water. It's going to go from a high concentration of water to a low concentration. We just tag on a different word to this special type of diffusion, and we call it osmosis. And it does it passively, no active. Okay? Now, let's talk about reabsorption in general, what's happening. All right? You see this whole thing. Make sure you know this. Draw it out 2 times a day until the exam. All right? Just make sure you know all the parts of it. At the PCT, proximal convoluted tubule, there's reabsorption going on there. We're going to be actively pumping sodium into the tubule. Okay? And this pumping of sodium is going to drive the reabsorption of other things. As I was explaining to you with the doorway, right? Now, 65% of all your reabsorption is going to take place in the proximal convoluted tubule. All of it. Except for glucose and amino acids. That's not 65%. In a healthy person, 100% of glucose and 100% of amino acids gets reabsorbed at the PCT. Okay? Cations, in fact, soluble substances are done through diffusion at the PCT. And organic nutrients, again, organic, anything with a carbon in it, okay? And selected cations by secondary active transport, which is secondary active transport is that thing I was explaining to you about with sodium. All right, it's using the power of sodium to allow things to get into the bloodstream. Okay, not ATP. It's using sodium's power, not ATP power. That's what we call secondary active transport. Okay. So if 120 milliliters of fluid enter the PCT per minute, then we have, as we talked about before, 40 milliliters will enter the loop of Henle, because 65% gets reabsorbed at the PCT. Okay? So this is just showing a picture. Look at it on your own time. But basically, you got here. Here's the filtrate. So this is the lumen of the tubule. Here's the blood vessel over here. So the peritubular capillary. And they're showing you the cells, and they're showing you how things are coming in. See, as sodium comes in, it's going to drive the glucose amino acids and some ions this way. It's using the power of sodium to get reabsorbed. And here's that thing where I was telling you about the na-na-na-na song, right? 3 sodium go out and 2 potassium go in. Okay? So you can look at this, and we'll talk a little bit more as we go along, but it's one of these things you just have to look at for about 10 minutes or so, and you could probably come up with some questions I would come up with on that. Okay? All right, now loop of Henle, right? We talked a little bit about this. This gets a little bit more complicated now. Reabsorption through the water gradient, going from a high concentration to a low. The descending part is going to reabsorb water. All right. So the descending part, through osmotic pressure, there's going to have water is going to get reabsorbed, making more of a concentration of the filtrate. So let me just do a very simplistic view of this. Should all look familiar, right? I just made it very simplistic. So what's happening here is that in the loop of Henle, water is going to come out, but all those electrolytes, and I'm just going to abbreviate by saying sodium chloride, but a lot of other things, that does not get reabsorbed. So as this gets further down to the hairpin turn, water is coming out, getting reabsorbed. The sodium chloride and all those electrolytes are not. So it's going to make the filtrate more concentrated or more dilute? Concentrated. Okay? And that's, if you have a long loops of Henle here, that's going to contribute mostly to making your urine very concentrated. Okay? The ascending part, again, we still have water in there. We still got all those electrolytes. But in this case, as it comes up, water is impermeable. It can't let water get reabsorbed. So water stays in there, but the sodium chloride does go out. So as it goes up, the ascending, does it become more, the filtrate, does it become more dilute or more concentrated? Good. All right, does that make sense? Not too bad. And this is just showing the same thing. Here's descending and here's ascending. We'll look at that and read that on your own. Okay? Now, we'll get back to this because we're going to talk about the blood vessel going here, the countercurrent mechanism. I'm going to finish up this reabsorption for the moment being. Reabsorption of DCT. Calcium, sodium, potassium, water. A lot of things are also going to be here. But most of the reabsorption has already taken place. 80% down here. We got about 19% that it still has to do between here and also over here. Okay? And remember that what targets here is aldosterone. Okay? Which means sodium is going to get reabsorbed, water is going to follow, the blood volume increases, blood pressure increases, but the charge also becomes more positive, so we have to lose a positive charge. So aldosterone is not only going to reabsorb sodium, it's going to secrete potassium to make sure that we add a positive charge, we have to lose a positive charge. Be careful of the wording too. If we have a lot of aldosterone here, we could say that we're going to increase reabsorption of sodium. But is that saying the same thing as that we're going to decrease the secretion of sodium? You see what I'm saying? It's going to lead to the same thing. So be careful with that. Collecting ducts. This here, what's going to target here is ADH. Water gets reabsorbed there and something called urea. Through osmotic pressure, we're going to reabsorb sodium into the bloodstream. ADH gets released, and then we also will have water reabsorption happening here. All right, let's talk about this urea recycling. It confuses people too. Urea. There's uric acid and there's urea. Uric acid is a byproduct of nucleic acid metabolism. Ooh, nucleic acid. RNA, DNA, that kind of stuff. We'll break down that stuff. And what releases is this thing called uric acid. 100% of this uric acid gets reabsorbed from the filtrate. It goes back into the bloodstream. Okay? We also have something called urea. Oh, and 10% gets secreted and excreted. So 100% gets reabsorbed. We also have some that's going to get secreted and go out here. So we still lose about 10%. But we also have urea. And this is a byproduct of amino acid metabolism. Okay? If you increase the proteins in your diet, you're going to increase urea. Proteins, well, when you break down proteins, you make amino acids, right? Amino acids make up protein. So if you have a high protein diet, you're going to increase your urea because we have to break down the amino acids. 50% of urea is reabsorbed and 50% is excreted. So it's going to go out. It's kind of a weird thing. 50% of urea, 50% go out your urine, 50% urine. But we also have 50% getting reabsorbed. We don't really need this byproduct. A lot of it, yes, we're going to have problems when it goes, a lot of it gets reabsorbed, it can cause a lot of problems. Ammonias get high in there, goes into our brain, we won't be able to, we'll go into a coma and we'll die. A small amount, we could actually utilize this, and I'll show you how. It's reabsorbed for this driving force. We had sodium that was a driving force for many things, but urea is going to be the driving force to reabsorb water in the descending loop. See, this is what's happening. See how this water is coming out. Well, why is the water going out? Water goes where there's more of a concentration to dilute it. So what's happening here is that, and I'll do this in purple, this urea is going to actually get reabsorbed to this area here. So now we're concentrating this area over here. Okay? The bloodstream. We've got a blood vessel going there. So we have more of a concentration out there than inside here. Because of that, water wants to go where there's more solutes to dilute it. So this, we're using the property of urea, not urea itself, but the property of just being another thing to cause a concentration to go up. We're going to put it here, and water wants to dilute that area. As it's diluting that area, it's taking it from the filtrate and making it more concentrated for the urine. See how that's working? So we're taking this urea and we're recycling it. It doesn't really go into the bloodstream throughout your whole body, but it stays in this area here. And it acts as the driving force to allow the water to get reabsorbed at the descending loop of Henle. Does that make sense? Can you visualize that? All right? You can read this on your own, but it's showing that whole thing where it's going back in here. There's the bloodstream. All right? I didn't draw a bloodstream here, but it really goes, it gets reabsorbed in the bloodstream. But it stays in that area, allowing the water to reabsorb. Okay? And it's showing the same thing over here. Here's urea as it's trying to concentrate itself. Okay? You understand the purpose of urea recycling? Yeah? Okay. Let's talk a little bit about secretion. The tubular secretion, which means it's going to go from the capillary into the nephron, right? It's into the tubule. So this moves metabolic byproducts, drugs, molecules that normally produced by the body into the tubule of the nephron. It can be active transport or it can be passive. Ammonia is produced by the epithelial cells of the nephron and diffuses into the lumen. We don't want high levels of ammonia to get into our bloodstream. Ammonia will get into the brain, as I explained to you before, put you in a coma. It starts off as forgetfulness. You can't recognize faces. It's very hard on the family if someone has, let's say, renal failure and they start building up their ammonia in their body. But it's a peaceful way to go. The kidneys, and we'll talk more about kidney failure, but kidney failure is a peaceful way to go. It's kind of like your body can't get rid of all the ammonia out the urine, so it builds up in your body, and you go into this loopy world. Does that make sense? I mean, I was going to go, I want renal failure. All right? Because it's a peaceful way to go. It's hard on the family because the patient doesn't recognize faces. Oh, hi, Gilda. Gilda, I'm your husband. You know what I'm saying? It's hard on the family. And also, hydrogen, potassium, penicillin actively gets secreted into the nephron also. So the PCT, this is where hydrogen and ammonia, that's what ammonia is NH4+. Here's the other side of the acid-base balance. This is where it all takes place. We did this reaction over here. CO2 plus H2O yields carbonic acid, and that's an intermediate, but then it turns immediately to a hydrogen ion and bicarb. Now, how do you get rid of carbon dioxide? Gas. How do you get rid of a gas? What organ? Lungs, right? So your lungs can get rid of that. But if this starts building up over here, if your lungs can't get rid of it all, then you have another organ to get rid of water-soluble things, ions. And what organ can get rid of this? The kidney. This is why I put the test, or your exam, the kidney, or the urology and respiratory on the same, because they have a lot to do with each other. You wouldn't think that, but it does. So this is the other side of the acid-base balance scheme. Lungs took care of this, and this is where the kidney takes care of the ions. And you can see where this is going when we talk about acid-base balance and electrolytes and fluids, which is the next chapter, and that's why I put this all together in one exam. They all have to do with each other. Okay? So we have this carbonic anhydrase that's in the PCT, proximal convoluted cells that do this. Okay? So what's happening here with the acid-base buffer, all right, if blood gets too basic, just think about it. If blood gets too basic, meaning that there's going to be a high pH, then you want to make sure that the kidneys can reabsorb hydrogen, right? If we have this, erase this Now this reaction, I know it's up here, but I think it's easier for you to. CO2 plus H2O. If I'm writing this often, I would know it for the exam. You might have to represent it by writing it out too. Who knows? But it's a pretty darn important reaction, right? So what's happening here is that if your blood is too alkaline, too basic, then your kidney wants to be able to reabsorb that, get that back into the bloodstream. When you put more hydrogens in the bloodstream, what is that going to do to the pH? Come on, guys. It's going to make it go down. So that's what's happening with this, is that we could do things here, which is pretty rapid. You could blow off a lot of CO2. This is the balance, right? It's a teeter-totter, as I explained to you. We could get rid of the CO2 quite rapidly by breathing it out. But it comes to, you have a cap, you can't do it. This is the best way to get rid of it, because it really just either goes right in the bloodstream or you pee it out. But this takes a long time to do. This takes hours. This takes like seconds to minutes. We're going to talk about acid-base balance later on, but you've got to see where we're going with this. So if this goes in, you're going to put more of this out. It's going to make your urine more alkaline. Does that make sense? Because we're going to reabsorb the hydrogens to put back into the bloodstream. That means the bicarb is going to enter. There's going to be a lot more bicarb entering your urine, which is going to make your pH of your urine increase, right? More alkaline. If the blood gets too acidic, then we're just going to reabsorb bicarb. And we'll secrete this. Or we'll take some of the hydrogens from the bloodstream, also secrete through here. And if that happens, then your urine becomes more acidic. Right? That's why we do urine dips, and we have an idea of what's going on with the person's blood by looking at the urine. Okay? So just, you do this on your own, but there's our Ca, is the carbonic anhydrase. All right, the enzyme that does this whole reaction that I just drew for you, and that's what's happening here. Here's the lumen of the lumen of the tubule, and here's your bloodstream. So it's doing this whole thing. So you just look at that and put it all together. Now, let's get back to this loop of Henle thing and talk about how the countercurrent mechanism goes on. I guess the pictures are up here, it makes more sense. It's hard for me to draw it, but we got this. Countercurrent mechanism. Fluid goes down like this, fluid goes up like this. Countercurrent, when you put them against each other. You see it? All right? Now the loop of Henle itself is countercurrent because of that. The descending is going one way, the ascending is going the other. We call this the countercurrent multiplier. So the loop of Henle is a countercurrent multiplier when you put the two limbs together here. The descending limb is going to be permeable to water, so water can come out, but this side it can't. And that's what the ascending is. All right, so it can't do that. So the collecting ducts in the deep medullary regions, down deep over here, are going to be permeable to urea, as I explained to you before.
Hiya! Welcome to Biomed Sessions with me, Ruse. Today we're going to be discussing the loop of Henle and the way it helps to concentrate urine in the kidneys. The loop of Henle is found between the proximal convoluted tubule and the distal convoluted tubule in the nephron. There are actually two types of nephron in the kidney, cortical and juxtamedullary. This happens to be a juxtamedullary nephron because of its long loop of Henle and the fact that it dips down into the medulla of the kidney. This type is well suited to the role of helping to concentrate urine. As discussed in my function of the nephron video, filtrate moves through the tubules and eventually exits the collecting duct as urine. But what we want to know is, why does the concentration of filtrate increase as you go further down the loop of Henle? Osmolarity is the concentration of a solution, often expressed as milliosmoles per litre. Filtrate in the proximal convoluted tubule has an osmolarity of 300 milliosmoles per litre, the same as the surrounding interstitial fluid. This means that the filtrate is isoosmotic with its surroundings, but it's a very different story in the loop of Henle. Let's zoom in. Let me give you a brief recap of what happens here, but if you want more details, including information about permeability, do go back and watch my previous video. Okay, so in the thick ascending limb, sodium ions are pumped out and negative ions such as chloride follow, making the medulla a quite concentrated and salty region. Water leaves passively from the thin descending limb because of the surrounding salty environment, and as the water leaves, the filtrate in the descending limb becomes significantly more concentrated. In addition, the salty medulla is also a major reason why water is able to move passively out of the collecting duct and be reabsorbed back into the blood, thus leaving behind more concentrated urine. But let's pretend that the filtrate is entering the loop of Henle for the very first time. Naturally, because the filtrate is coming from the proximal convoluted tubule, it will have the same 300 milliosmoles per litre osmolarity and be isoosmotic with the interstitial fluid. However, this situation is not ideal for creating concentrated urine. So in the latter part of the loop of Henle, our aim is to create a difference of 200 milliosmoles per litre between the ascending limb and the interstitial fluid. The only way to do this is by pumping out sodium ions. So let's pump. As you can see, the osmolarity in the ascending limb has decreased due to a loss of sodium, whereas in the interstitial fluid it has increased due to a gain in sodium. And if you look closely at the values, you will see that we have achieved the 200 milliosmol per litre difference, otherwise known as a gradient. But what's happening with the descending limb? Here the filtrate needs to equilibrate, meaning that the water will leave passively until the filtrate in the descending limb reaches the same osmolarity as the interstitial fluid. Time to equilibrate. Note, equilibrating does not change the osmolarity of the interstitial fluid. Filtrate moves through the nephron continuously, but I'm going to break this down into steps for you. Let's advance the fluid in the loop of Henle. When brand new 300 milliosmol per litre filtrate enters the descending limb from the proximal convoluted tubule, the filtrate already in the loop of Henle is pushed further along. Bringing back our values for the interstitial fluid, we see that our gradient has been messed up. So we need to re-establish that ideal 200 milliosmol per litre difference. Let's pump out some sodium ions. Okay, although the values vary, there is still a difference of 200 at all levels, so we're content. Now to equilibrate the descending limb with the interstitial fluid and advance the fluid in the loop of Henle a little. Lo and behold, the gradient is messed up again, but we know what to do. Let's pump and establish that 200 milliosmol per litre gradient. Equilibrate to match osmolarities and advance the fluid. Okay, one more time to get the point across. Our gradient is messed up. Pump out sodium to restore the gradient and equilibrate. Alright, so looking at our values for the interstitial fluid, we can see that the osmolarity increases the deeper you go into the medulla. As all that pumping and equilibrating continues, we eventually reach a maximum concentration of around 1200 milliosmoles per litre. The entire process that I have just described is called countercurrent multiplication. Countercurrent because the filtrate flows in opposite directions in the limbs of the loop of Henle, and multiplication because this countercurrent flow enables the effects of the gradient to be increased, i.e. multiplied. So in summary, the loops of Henle of juxtamedullary nephrons are involved in the process of countercurrent multiplication, which enables the interstitial fluid to become more concentrated, i.e. increase in osmolarity the deeper you go into the medulla. The high concentrations in the medulla facilitate the passive movement of water out of the collecting duct and result in more concentrated urine. Okay, hopefully this tutorial was helpful to you. If so, don't forget to like and subscribe, and do drop by my Facebook page to get the latest updates on upcoming videos. All links will be left in the description. Okay, see you in another video. Bye!
Then we have the countercurrent exchanger. Now, in actuality over here, where it's difficult for me to draw. But the way, and I'll show you the next picture over, it'll make more sense. But the way that this efferent arteriole is, it kind of rides along here, it goes this way, like this, and goes up like that. I know it looks weird. But when you follow the bloodstream, it's going this way, and it's going this way. You see it? So and like I said, this is all peritubular capillaries, but when we deal with the capillaries next to the loop of Henle, this area here, this is called the Vasa Recta. It's still capillaries, but has a special name to it. But we have another countercurrent mechanism going on between the loop of Henle and the Vasa Recta. This is going down, this is going up. This we refer to as the countercurrent exchanger. And in a way, don't want to get too detailed with it, in a way, it's just being able to deliver the blood to the cells in that, well, it maintains the osmotic gradient. And the blood vessels here, we have little blood vessels that actually give blood supply to the cells themselves. It's kind of like the vasa vasorum to the blood vessels. Vasa recta will do the same thing too. So it's showing you over here. See how like it comes up, all right? So the descending limb is here, but descending limb of loop of Henle is here, and here's where the blood is coming up. So there you got this. So the blood, what's happening here as the water is coming out and going into the bloodstream, it's making the blood get more dilute, but it's making this, the filtrate, more concentrated as we go up this way, all right? But I just want to show you they go in countercurrent areas. The countercurrent multiplier is the loop of Henle. The countercurrent exchanger, because you're exchanging it into the bloodstream, that's where the loop of Henle is compared to the Vasa recta. Okay?
Are you ready to have your mind blown? Of course you are. Your kidney is phenomenal. Look at this. Okay, we know that in the interstitial fluid, we've got this crazy concentration gradient. And the question is, as we're moving solutes in and out, and as we're moving water in and out, and changing the concentrations of the filtrate, how do we pick that up in the bloodstream? And it has to do with this amazing setup called a countercurrent exchanger. So watch. We know that the filtrate is coming in at 300 milliosmoles. And you can see that I'm back to our original drawing. I've got the fluid in the proximal convoluted tubule, and it heads down into the descending loop of Henle. As it heads down, we know in the descending loop of Henle, we're going to send water out. That's going to concentrate the filtrate. The filtrate is going to get more and more concentrated, and we've done this, so you're cool that we keep getting water out the descending loop of Henle. It's going to keep coming out and out and out until we reach the base where now we've gotten rid of so much water, but now that water has gone into the interstitial space. Now we're going to pump solutes out. So I'm telling you that our solutes are getting pumped out now. Now, here's the cool part. As we pump out our solutes, you're awesome and accept the fact that we keep pumping until we get like, what, 100, whatever. Here's the amazing thing. Our blood also has an osmolarity. Do you agree with that? It's going to be 300. But look at what happens. Look at who the blood goes by first. The blood is passing by right next to the ascending loop of Henle. So literally, the solutes that come out go into the ascending, the blood supply. And there's a concentration gradient because now all this stuff is coming out and it's going to be picked up by this lower concentration blood that's flowing in. So the concentration of the blood is increasing as it goes down because it's going right next to the ascending loop of Henle, and it's picking up all those solutes that are being pumped out. So does that work for you? Can you imagine how that is the case? What happens if the blood picks up the solutes and takes it out of the interstitial fluid? We now don't have that 200 gradient anymore, so we just pump more solutes out. So this is really a pretty endless situation where we can continue to pick up solutes until literally we reach 1200. Now, this is another incredible thing that happens. As we ascend, now what was coming out of this side? Doggies of a feather. It was water. Seriously? So as we ascend in the backward direction, because it's a countercurrent exchanger, water is going to come out. We're going to keep going up and pick up the water. So as the water gets picked up, our concentration is going to decrease until we get back out of the madness at the concentration that we started. Meanwhile, we picked up solutes. We didn't lose anything. All we did here was grab stuff. We grabbed solutes on this side, and we grabbed water on this side. And by grabbing solutes first, we were able to pull them all out. And by grabbing water second, we were able to pull it all out and take it back into the blood supply. Does that blow your mind? That is how we ended up filtering out 180 liters of fluid and then reabsorbing all but 1.5 liters of PP. That's unbelievable. We're going to use our medullary concentration gradient in the next section when we talk about, in the next lecture, when we talk about how hormones help us maintain osmotic equilibrium. But we're not going to deal, again, with how it's maintained or how it is established. So it would not surprise me in the slightest if you needed to go mess with this again and again. I can't tell you how many hours I've spent messing with this. It's definitely a tricky concept. So spend some time, share any resources that you find that are awesome, and kidneys part 2, cha-ching, kidneys part 3 coming up.
Questions on that? And you can see it all here too. Okay? Now, let's talk about regulation of urine concentration and volume. This is easier to manage. Osmolarity is a fancy word for concentration. The number of solute particles dissolved in 1 liter of water, that's what we call osmolarity. It's how much, it's how concentrated is that? Do you take your lemonade and you have your lemonade again is water and crystals? How much of the crystals are in that water? What's the osmolality? Is it very concentrated? Is it very dilute? And this will reflect the solution's ability to do osmosis. Remember, If it's very concentrated, water wants to go where there's more solutes, more of the particles. To dilute it, you take your lemonade, you put, let's say, I don't know, you have a bunch of crystals in there and only 1 cup of water. It's going to taste very bitter. So it's going to want you to put water in there. So in an area it's very concentrated, is going to allow water to get into that area through osmosis. Okay? Which we've been talking about with oncotic pressure. Okay? So that's what's going on here. So body fluids are measured in what we call milliosmoles. That's how we talk about concentration. There's a unit to it, and it's milliosmoles. And kidneys keep the solid load of body fluids at about 300 milliosmolalities. All right? And this is accomplished by this countercurrent mechanism, which I really don't want to go into the details with it. I would need an extra hour to explain that. Okay? So the interaction, again, is this countercurrent multiplier and the countercurrent exchanger. The multiplier is the flow filtrating through the loop of Henle, one going down, one going up, and then you have the countercurrent exchanger, which is between the loop of Henle and the Vasa recta. And the solute, anywhere, depends on how concentrated it is, but you want to keep it about 300, but when you have these loops of Henle really deep, they can go up to an osmolality of 1200. They get more concentrated. Okay? It equilibrates. So I think that, yeah. So when you look at this is our kidney. This area here, where the cortex is, it's going to be about 300 osmolalities over there. But if you have loops of Henle that go really deep down into the medulla, really deep, they could go down to they could go up to 1200 osmolalities. So it's going to be more concentrated deep in the medullary cavity, or no, not the medulla, but the medulla of the kidney. Right? You would think that, as I explained to you, if you have long loops of Henle going down, they're going to make you have more of a concentrated urine. And that's what it's trying to show you over here. Okay? To form a dilute urine, it's diluted in the ascending loop of Henle. So as you go up, it's going to become more dilute. Remember, water, it's not going to be able to leave, so it becomes more dilute there. And we also have ADH. Remember, over here, we have another opportunity where we can dilute the urine. Because ADH, if this floods with ADH, what happens to the concentration of your urine? What does ADH do to water? Reabsorbs water. So what happens to the concentration of your urine? It becomes more concentrated. Does that make sense? If this area doesn't have much ADH, then more water will not get reabsorbed, so it stays in your urine, and it's going to make more of a dilute urine. Okay? So the collecting ducts remain impermeable to water. All right, no further water reabsorption occurs, except if we have ADH. Right? ADH is going to allow water to get reabsorbed at the collecting ducts. Sodium and selected ions can be removed by active and passive mechanisms, and the urine osmolarity can be as low as 50 milliosmolarity, depending on how much of the ADH is out there. Like your urine can be very, very dilute. Okay? So forming concentrated urine, again, the ADH is going to take part in that, right? If it's going to be loaded with this area of ADH, it's going to get absorbed, which is going to make the urine more concentrated. All right? It's all about that. ADH dependent, so it's all about ADH. Pretty self-explanatory on this stuff. All right? So in an area, just to show you, in an area that has low ADH, so here's your collecting duct, here's your loop of Henle, ECT, collecting duct. If we don't have much ADH around here, then what'll happen is water will stay in here and go out your urine. So the concentration will stay the same. But if you have ADH in this area over here, right, it's going to reabsorb the water, which means there's not going to be much water going out your urine, but your electrolytes will still be there, which means you're going to have a more concentrated urine, 1200 and so forth.